Hello there, my name is Kyle Thompson, and I'm a member of the Human Performance Lab over at the University of Guelph in Ontario, Canada. And today I'm going to be going over the presentation that I gave for my PhD defense, looking at the influence of blood flow restriction training on maximal aerobic capacity. I was lucky enough to be under the supervision of Dr. Jamie Burr, and you can see in the middle of the screen some of the other universities that we collaborated with, and down in the bottom left corner some of our funding agency, and down in the bottom right some of the logos that we actually made for some of the research studies that we completed. Those are the BEATS logos there, and then we also were lucky enough to partner up with Rowing Canada for one of our final studies here. So what I'm going to be going over today is some of the research that I was able to conduct, and just for simplicity and time to keep things as concise as possible, I'll be just reviewing the key findings from each of the separate research studies. So to follow along today, there's two key concepts that you need to be familiar with. The first is this idea of maximal oxygen uptake, or what we call VO2max. And the second is this idea of blood flow restriction training. So first, let's focus on our VO2max. So what we know is that with increasing work intensity, so if you're cycling against a greater resistance on the bike, while you're running faster outside or on the treadmill, we know that the amount of oxygen being consumed to help generate energy in the form of ATP also increases. So when we're talking about our VO2max, or our maximal oxygen uptake, what we're really interested in is what I've got highlighted here at the end of the exercise test, where there's an increase in work intensity, but there's a plateau in the amount of oxygen being consumed. So there's no longer a greater oxygen utilization, even though we're working harder. So this is our VO2max here. The way that we bring in oxygen into the body and use it begins in the air. So first, the air that we breathe in is approximately 21% oxygen, and we bring that into the lungs, where it diffuses through the lungs and binds to the hemoglobin protein in our red blood cell, which acts as our major oxygen transport system throughout the body. This red blood cell then goes from the lung to the heart, where we can pump it to the working muscles in exercise. And we call this our cardiac output, and we measure this in liters per minute. So the amount of blood, the volume of blood that we can pump out, is our cardiac output. The blood itself and the oxygen carrying capacity of the blood is largely dictated by the hemoglobin mass concentration, or the hemoglobin concentration in the blood, or the amount of red blood cell that we have in the blood. And the other thing that's really important here is when the blood is going to the active limbs through the major arteries, the total amount of blood flow that we can get to is another very important characteristic, allowing us to transport oxygen to the working muscle. Once the red blood cell and the oxygen get to the skeletal muscles, we can then offload the oxygen where it can be used in the mitochondria to help generate ATP, or adenosine triphosphate, which we can use for things like calcium handling or skeletal muscle contraction. So now that we have a rough idea of how we bring oxygen into the body and transport it around, we should be familiar with several major determinants of our VO2max. And the first I'd like to point out is our cardiac output. So the total amount of blood that the heart can pump is tightly related to our maximal oxygen uptake. Similarly, we see that the total amount of hemoglobin that we have, and we see this similar relationship between the total amount of red blood cell or the total amount of blood volume with VO2max, also is a strong relationship. And the last major determinant that I want to pull your attention to is, like I said before, the total amount of blood that you can get into a working limb. So the arterial blood flow has also a very tight relationship with the VO2max of that limb. So these are the three physiological characteristics that are most likely to influence VO2max. So now we can move on from VO2max and we can explore this idea of blood flow restriction training. My supervisor, Dr. Jamie Burr, posed for the photo here while performing a resistance exercise, and he's got the BFR bands around his upper thighs there. So what we see during normal exercise is that in the red arteries and the blue veins, we have an increase in blood flow to help deliver oxygen and nutrients to the working muscle. And on the venous side in blue, we're able to help clear metabolites, or things that the muscle is releasing that may limit our ability to produce skeletal muscle force. What we see with the BFR exercise is when we inflate cuffs to approximately 160 to 220 millimeters of mercury, which is probably about the peak pressure that you'd experience if you're going to go to the doctor's office and get a brachial blood pressure measurement around the arm. We see that the vasculature can be slightly compressed, and this reduces the rate of flow into the muscle and the rate of blood leaving the muscle. So this reduces the rate at which we can deliver oxygen and nutrients and also clear metabolites. This type of exercise, known as blood flow restriction training, has been commonly researched, and there's a number of studies you can see listed on the side here, looking at how much this type of training can induce muscle hypertrophy, and typically it's compared to heavy load resistance training. And what we see as a general consensus is that if you perform light load resistance training with blood flow restriction, it gives you the same degree of hypertrophy as heavy load resistance training. So if we move on here and we combine these two concepts that we're just familiarized with, we can see that we come up with something a little special. And maybe we call this something like BFR endurance training, where instead of performing low intensity resistance exercise, like you saw in the previous slide, here we're performing low intensity endurance exercise, such as walking, but with the cuffs occluded around the upper thighs. So we're not the first study to have done this, or the first group to have looked at this. A study by Park and colleagues in 2010 saw that with collegiate level basketball athletes, there was a large increase in VO2 max after performing walking training, like you just saw in the video, 2 times each day for 2 weeks. And it's important to point out that there was this improvement in 2 weeks, because typically if we want to see an improvement in VO2 max in this short a time frame, we either need to do a lot of exercise volume, or the intensity of the exercise volume needs to be quite high. Also impressive, in a more recent study here by Held and colleagues, we see that there's also an increase in VO2 max in highly trained rowers. And just to show you how trained the group of athletes were in this more recent study, I've plotted both the Park and colleagues and the Held and colleagues research data on the same y-axis. So you can see now they're comparable to each other. And essentially, when there's a relative VO2 max over 60, especially in quite large athletes, we consider them to be quite aerobically trained. And despite that initial training status being so much higher in this more recent study, we see that the two studies by Park and colleagues and Held and colleagues have relatively similar increases in VO2 max after BFR training. Now, despite this, it's currently unclear what adaptation is responsible for the increase in VO2 max that we see, which leads to the overall purpose and aim of my thesis, which was to determine what physiological adaptation increases VO2 max after low-intensity BFR training. And in trying to answer this question today, we're going to look at some acute responses to BFR exercise. We're going to do a few training studies and look at training adaptations, and then we'll apply this in a more practical approach. So first, if we want to focus on the acute responses to BFR, in our first study here, our first objective was to characterize the blood volume distribution in the body during BFR exercise. And our second objective was to look at blood volume regulating hormonal response to BFR. So the reason we wanted to do this is because we know if we cause blood volume redistribution away from the core of the body, which is what typically happens during intense exercise or perhaps exposure to the heat, or if we're going more experimental, during lower body negative pressure, we can have the release of blood volume regulating hormones, which act on the kidney to cause fluid retention, which can cause blood volume expansion. And remember, this could be important because we know there's a tight relationship between the total amount of blood that you have and your VO2 max. So for this first study, we essentially did 3 mini studies using carbon monoxide rebreathe techniques, echocardiography, and collecting blood and analyzing it for specific hormones involved in blood volume regulation. And we believed that blood volume in the central compartment would be redistributed to the areas performing the BFR exercise, and that when you perform BFR exercise, there'd be the release of these hormones involved with blood volume retention. So on a first day, you have to have your supervisor. It doesn't work if you don't. We had him perform a carbon monoxide rebreathe test, which essentially is you rebreathing a small amount of carbon monoxide that acts as a labeling agent on your red blood cells so that you can calculate the total amount of red blood cells that you have and also the total amount of blood volume. On a second day, we performed an acute bout of the BFR treadmill walking protocol and also, with the cuffs occluded, again performed the carbon monoxide rebreathe technique. So what we have after day 1 is a total body blood volume, and after day 2, we have a blood volume of we have a blood volume above the level of the cuffs. And the reason we have a blood volume above the level of the cuffs is because the carbon monoxide that you've inhaled is unable to mix with any of the blood below the level of the cuff because you have completely occluded the vasculature. So what you can do with these is you can subtract them from each other, and then you can determine that approximately 25% of the total blood volume of each individual is trapped below the level of the cuff. In addition to this, we performed an exercise test on our echocardiography table, which you can see on the left there. And while the participants are cycling at a relatively low intensity of 60 watts, we're using echocardiography or ultrasound of the heart to measure the heart dimensions. And what we can see is when the exercise intensity is the same, but we include BFR, there's a slight reduction in the end diastolic volume, or the total filling of the left ventricle. So taken together, when we see that there's a reduction in heart filling, and we see that there's a large amount of blood that can be trapped below the level of the cuff, we can therefore assume that there's blood volume is being redistributed away from the core and to the limb that's performing the BFR exercise. Next, we need to confirm whether or not blood volume regulating hormones are being released. So to do this, we collected blood before and after the walking protocol that was used by Park and colleagues and previously showed an increase in VO2max. And in short, what was done is we performed 5 bouts of walking exercise for 3 minutes with the cuffs inflated. And there were 1-minute rest periods in between these bouts where the cuffs remained inflated. However, halfway through, we deflated the cuffs just because it was quite uncomfortable for the participants. Now, when we measure renin and copeptin, which are both hormones involved with blood volume regulation, we can see that after the BFR walk versus the control walk, that there's a significant increase in these hormones in circulation. What's important to know from another study that we conducted is that we actually see a blunting of this effect after training. And it's important to note this because typically during exercise interventions, if we see a blunting in a stress response to an exercise, it could be a clue that there hasn't been an adaptation responsible for that blunting. So perhaps the exercise stress is no longer as stressful. So to summarize our findings from the first study here, we see that BFR exercise reduces central blood volume due to blood pooling in the occluded limbs, and we know that BFR elicits a hormonal response that suggests it should cause fluid retention, which would lead to an increase in plasma volume. So moving on now to look at what happens when you perform BFR repeatedly in a training environment. So building on the work that I just showed you, we're now going to look to see whether or not blood volume is expanded after BFR training. And just to remind you again, there's a tight relationship between VO2max and blood volume. So if we see an increase in blood volume here, that could be the reason why past research studies have seen an increase in VO2max after BFR training. Our first objective was first to confirm what others have seen, that indeed BFR training does increase VO2max, and the second objective is to determine whether hemoglobin mass or total blood volume increase after this type of training. To do this, we got 28 recreationally trained participants and split them into 3 different groups, a control, a low volume BFR group, and a high volume BFR group. And we believe that with greater BFR exposure, would result in greater increases in VO2 max, and we thought that an increase in hemoglobin or blood volume would be responsible for those increases in VO2 max. So here's our study timeline. You can see weeks across the top there. And along the left side, we've got our control and BFR high volume groups and our low volume group at the bottom. So for our control group, we did the walking protocol that we'd performed before, and that was done by parking colleagues. We'd perform that 2 times each day for 4 weeks. The high volume BFR group also did this walking protocol for 2 times per day for 4 weeks, but of course with the cuffs inflated around the upper thighs. And the low volume group performed the same walking protocol, but only 2 times each week, but for 6 weeks. Of course, you have to have your supervisor again perform the CO rebreathe, and along with all the participants at the beginning and end of the training protocol. And we measured VO2 max repeatedly throughout the training protocol to get an idea of time course. So first, if we look at our VO2 max results for our control group, we can see that there's no changes over the 4 weeks. So what this suggests, and what would make sense, is that walking 2 times a day on the treadmill is insufficient of an exercise stimulus to cause any changes in VO2 max. In line with what was seen by Park and colleagues, after 2 weeks of BFR treadmill walking, we can see an increase in VO2 max. And we can also see that if you perform the low volume BFR training, there's also an increase in VO2 max. So taken together, we can conclude from here that both BFR protocols increase VO2 max, but over different timelines. Now looking at the hemoglobin mass, and this is also the same findings for blood volume, we see that there's no changes after the control training, which we would expect because there was no change in VO2 max. Interestingly, we saw no changes in hemoglobin mass or blood volume after the low frequency BFR protocol, but we also saw no changes in hemoglobin mass or blood volume after the high frequency BFR protocol. So therefore, there was no relationship between any of the increases in VO2 max that we observed and any changes in hemoglobin mass or blood volume. So to summarize this, we know that both low and high volume BFR training can increase VO2 max, but over different time frames. And we know that that increase in VO2 max is not related to any change in hemoglobin mass or blood volume. So just to pull everyone back and give a little bit of context again, if this major determinant of VO2 max is not changing after BFR training, but we are seeing an increase in VO2 max, then it becomes more likely or more probable that we will see a change in maximal cardiac output or maximal limb blood flow. So we've got to do that. To do this, we went to a conference in Copenhagen and we listened to Dr. Jose Calve give a talk there where he went over some of his past research. And of course, I thought this was fantastic. So I messaged Dr. Burr and told him that we needed to do a collaborative project, to which he gave me a strong thumbs up. I took that as a full conviction that he was fully on board. So of course, 6 months later, we were flying 12 well-trained athletes from Canada over to the University of Las Palmas on the island of Gran Canaria, one of the Canary Islands, just off the west coast of Africa here. In the end, it was a very strong collaborative effort that we were able to conduct here with members from the University of Las Palmas, the Norwegian Institute of Sport Sciences, the University of British Columbia, University of Calgary, University of Limerick here, and of course the University of Guelph. So our primary objective for this study was really to determine whether BFR training increases maximal cardiac output, like blood flow, or because of the setup that we had, we could also look at oxygen extraction across the limb. And we recruited 12 highly trained participants and we split them unevenly into either the control or BFR group. So at this point, including our data, there had been about 11 studies that had shown increases in VO2 max after performing BFR exercise. So for this study, our focus wasn't necessarily to see whether or not there would be an increase, but really focus on what adaptation is responsible for that increase. So that's why we favored more participants into the BFR group, whereas we had the 4 participants in the control group to look at the reliability of the methods, especially after we're flying our participants halfway around the world and changing time zones. We hypothesized that BFR training would increase VO2max via either an increase in maximum cardiac output or maximal leg blood flow. So you might be wondering why we wanted to team up with this group. Well, of course, Dr. Calbay and colleagues are very famous for doing these catheterization prep. So you can see in this image, we've got two catheters in the femoral vein and one in the femoral artery. And the reason that this setup is important is because this setup allows us to use something called the thermodilution method. So this method involves us injecting a small amount of cold saline into the femoral vein. And I've got an image here to show you the different catheter thermistors here registering the temperature as we perform those injections. So you inject the cold saline in through the infusate catheter. So you can see in green here, there's a rapid drop in temperature. But what's cool is that the second catheter that's in the vein is also measuring blood temperature. And when that cold bolus of saline passes by, you can see that the rapid drop in temperature and recirculation of further warm blood causes a dip in temp. And that allows us to use that area above the curve there to calculate leg blood flow. Similarly, on the arterial side, the cold bolus goes up to the heart and then comes back down to the leg where the arterial catheter is placed, and that drop in temperature that we see allows us to calculate cardiac output. So also, because we've got the catheters on the arterial and the venous side, at any time point we can pull blood from both sides simultaneously, and we can measure the oxygen content going into the muscle and the oxygen content coming out of the muscle. So we can essentially look at how much oxygen the muscle is extracting. So once we've got that preparation done, which takes about an hour and a half to 2 hours, we then get our participants on the bike and we perform an incremental exercise test where we're performing gradually more intense exercise conditions on the bike. At each exercise stage, we're injecting that cold saline with a thermodilution method in duplicate, which you can see with the purple arrows, and we're taking a blood draw from the arterial and the venous side at each stage as well, so we can measure oxygen extraction. Now what you can see is in line with our previous findings and that by others. There's an increase in VO2max after the BFR training, which consisted of the walking that you've seen earlier. There was no changes in oxygen extraction after the training period. So the amount of oxygen that the muscle is taking up at a unit of time is not increasing. What's interesting is that we saw no changes in maximal cardiac output. However, we did see that there was an increase in leg blood flow, each leg of about 1.2 liters, so in total about 2.4 liters. And with that increase in leg blood flow, there's an increase in oxygen delivery because we've got our oxygen bound to our red blood cells there. And with that increase in oxygen delivery, we know that especially trained athletes are good at using that oxygen. So with that increase in oxygen delivery that we saw, there's also an increase in leg VO2max. So the leg is consuming more oxygen. So to summarize this, and this is really the main aim of the overall thesis, we see that systemic or whole body VO2max increases after BFR training due to an increase in maximal leg blood flow, leg oxygen delivery, and leg VO2. So there's a greater amount of the total blood being pumped from the heart going to the working limbs, which are the legs. Finally, moving on to our last study, applying this in a more practical approach, we partnered up with Rowing Canada, and our objective here was to determine whether BFR training improves time trial performance in elite and world-class rowers, and our second objective was to determine whether known determinants of rowing time trial performance also improve after training. We hypothesized that time trial performance would improve after BFR training and that VO2max and workloads that elicit specific blood lactate concentrations would increase after BFR training. Here's our study timeline here. So initially we had the athletes perform a 2,000 meter time trial performance on the rowing ergometer, and this is important because this is a common test used by the Rowing Canada team to determine athlete readiness. So the participants are very familiar with this, and we're not expecting there to be any large learning effects from performing the test multiple times. They performed a graded exercise test, just like what you saw we did previously on the bike. However, here we're doing it on the rowing ergometer, so there's gradually more intense steps. And at the end of each of these steps here, they're performing a blood lactate measurement so that we can develop blood lactate curves. We also use the CO rebreathe technique again here to get at hemoglobin mass and blood volume before and after training. But like you already know, and this confirmed this as well, in this population, there were no changes in hemoglobin mass or blood volume after training. The training for 4 weeks was a little bit different than the walking protocol that we'd used previously. Instead, for this study, we mimicked the training protocol that was done by Haldane colleagues that I showed at the beginning that also saw large increases in VO2 max. So for that study, they performed 2 bouts of 10 minutes of rowing at a relatively low intensity. And this intensity is at what we'd call a 2 millimolar blood lactate concentration. So just for reference, at rest, you would be at about a 1 millimolar lactate concentration, and during intense exercise, you might be around a 15 millimolar per lactate concentration. So rowing at a 2 millimolar lactate concentration is quite easy for these athletes. After the 4 weeks of training, all the same tests were repeated. So what we can see for the time trial here is that female and male athletes largely had a big increase in their time trial performance. However, what you might note here at the bottom is that there's no control group. So one of the nice things about working with this level of athlete is that they're extremely well motivated, and you don't need them to encourage them too much to put in a strong effort. They really want to be doing the best they can at all times. However, one of the challenges is that this athlete and the coaches for these types of athletes, they don't really want to be guinea pigs to see whether or not something works, but more so to see how much something works. So to that point, what we did is we took historical data from each of the athletes, which you can see plotted with the black lines, and this represents the expected or the historical variability from test to test. So it would be normal for them to have this amount of change from pre to post time trial test. However, what I think you can appreciate is that with the black dots and seeing that the improvement is shifted to the left, and it, just to orient you, a negative % change means that they're getting faster, which is what you want in a time trial test. It exceeds the historical variability. So it's outside the black lines, meaning that they had a greater improvement here than you would have expected. And what's also important is with the stars that you can see there, these are 8 athletes on the team that had all-time 2-kilometer personal bests. And 4 of those athletes were previous Olympians. So it's not just that the newcomers to the team are improving quite a bit with training. Just like with previous studies, we see that there's an increase in VO2 max, at least with the females. But with the male, we didn't see this increase in VO2 max. So another challenge with working with this population is that there's only so many of them. And for this test, we only had 8 athletes perform both the pre and the post testing. And what's interesting to point out is that one athlete at the top there had a large reduction in VO2 max. And unfortunately, we don't have the luxury of pulling them in for another training session. And we also don't have the luxury of just getting more national level rowers. What's interesting, though, is if we did remove them from the analysis, now we do see that there is an increase in VO2 max as well, which would be in line with what others have seen. Lastly, here we see that the workloads eliciting 2 millimolar and 4 millimolar blood lactate concentrations increase from pre to post training. So this suggests that the athletes are able to work at a higher intensity while still able to produce the same amount of lactate or clearing it more effectively. So both the VO2 max and the workloads eliciting these blood lactates are physiological predictors of rowing performance, and as we see, they largely both improved. So to summarize this, we see that time trial performance improved after 4 weeks of training, and that determinants of rowing performance also improved. To summarize everything, we see that during the acute responses to BFR, blood volume regulating hormones are released after BFR exercise. And when we do training, we see that VO2 max does increase. So that's not because of an increase in blood volume, but it's because of an increase in leg blood flow, leg oxygen delivery, and leg oxygen consumption. And lastly, when we perform this training with elite level rowers, the national rowing team in Canada here, we see that there's a large improvements in time trial performance. So before we wrap up the video, obviously there's a huge amount of help that's gone into this. The committee and examination members, thanks for always helping out and giving lots of good feedback and ideas about how we can best design the studies. My supervisor, Dr. Jamie Burr, he's obviously super influential in how we go about doing our studies, and he's very supportive. I guarantee that there's no other supervisor who would have agreed to most of the studies that I proposed. So without him, a lot of this research definitely wouldn't have happened. So definitely appreciate that. And then, of course, we've got a ton of lab mates, and Alana there turned lab mate into my wife, and we got our baby Juliet there in the middle, so we're super happy. And then we've had all our collaborators from around the world now, and helpers in the lab. So thanks so much to everybody. Really enjoyed spending time with the lab and being able to do the research in the human performance lab at the University of Guelph.